All right. So welcome to chapter 19. Chapter 19, we're going to review the heart. And this is another big chapter like the last one. So your unit test that's coming up with chapter 17 and 19 is a pretty big, a lot of information, a lot of note cards you may need as well. In this case here, we're going to talk about the heart. And the heart chapter 19, pretty sure it's chapter 19. All right. But first, again, this is not a comprehensive review. Okay, focus on the materials. By reading the chapters, you're responsible for all the information in the chapters. Um, I will try to help focus on some topics so that you can better get an understanding of where the test is going to go and how the questions are going to be, more or less. Okay? All right. But first, let's have some fun facts. And I'm not sure if you know any of this, but let's have some fun facts. Your heart beats about 100,000 times a day. 35 million times a year. During average lifetime, the heart will beat more than 2.5 billion times. Can you imagine that? 2.5 billion? That's a normal heart rate. Imagine if someone has a high heart rate, it's going to be a little bit more, right? So 2.5 billion times in an average lifetime. If you take a tennis ball, give it a good squeeze, you're using about the same amount of force your heart uses to pump blood out of the body, out to the body. Hopefully not out of the body. Even at rest, the muscles of the heart work hard, twice as hard as the leg muscles of a person sprinting. So take that tennis ball. Oh, I got one right here. What coincidence? Right, so if you squeeze it, right, that's pretty sturdy, pretty good resistance. But that's how much force is needed to eject. Right, that's your cardiac output to eject blood. We'll go over that later. And your body is about 5.6 liters, 6 quarts of blood. 5.6 liters of blood circulate through your body 3 times every minute. In one day, your blood travels a total of 12,000 miles. Right? That's 4 times the distance across the United States from coast to coast. Right? So 3 times a minute, you're pushing 6 quarts around. That's kind of crazy. Think about it, right? And lastly, that lub dub you hear, right? We know, and you should know this too from the readings, that lub dub that you hear is the closing or the snapping shut of your valves, right? Mitral versus tricuspid versus aortic semilunar versus pulmonary semilunar valves. So stuff that you should know, but there you go. Your lub dub is the opening and closing, most of the time the closing of those valves. Now, on to the review. So again, some areas to focus on, okay? Heart anatomy, basically the anatomy of the heart. Atriums, ventricles, who's inside, who are your heartstrings, what are they connected to, who are the papillary muscles, everything you learn in lab. There's a couple good videos this in Unit 2, so check them out. So know your heart anatomy and all the structures of the anatomy. Not much physiology per se yet, but I'll touch on that a little bit, but you should know your heart anatomy. Coronary arteries, location and name, where they arise from, and then of course, once they get to the heart, how do you feed the coronary arteries as you exit the aorta, right? And then they're gonna start branching. You have some on the ventral side, you have some atrial ones, you have some marginal branches, you have lateral branches. Basically, what I want you to do is know how the heart is supplied with blood and the vessels that are responsible for supplying the heart with blood. Action potential and calcium gates, be familiar with. Okay, well, this is pretty specific, but we know the heart works with action potentials. We also know it works with calcium. Right? Calcium is a major player in activation of neurons. It's the same thing with the heart itself. So be familiar with whatever the textbook talks about as far as calcium gates and action potentials. And you kind of go over some action potentials as you go through the systole and diastole of the heart, how hard it's working versus how hard or how well it's relaxing. So be familiar with those action potentials, right? The repolarization versus depolarization. If you're looking up on the screen, you see a cross section of the heart, and as my cursor comes down, the Purkinje fibers are in here, and we know that there's going to be a beginning depolarization down at the apex as that heart contracts from the apex up. And the reason for that, right? Think about the heart, and as it contracts, it just doesn't squeeze the blood out. It actually takes the heart, it squeezes and it wrings it. It's almost like taking a towel and you squeeze and wring, right? So that's why the depolarization occurs at the apex. So that it's like a tube of toothpaste. You're getting all the blood to eject down low here, and it's a contraction and a twisting, very subtle. pretty cool, actually. All right. Well, there is your pathway, right? There's your conductive system. AV node, actually it's SA node, AV node, Purkinje fibers, and so forth. Be familiar with those pathways. What is an SA node? What is an AV node? Be familiar with the SA node versus AV node. What if the SA node stops working? Okay, if the SA node stops working, then and the AV node takes over, then you're going to have some you're going to have different rhythms. So I'll mention that later, but you should be familiar with that as well. As you go through the anatomy of the heart, ask yourself, if this part of the anatomy failed, what is the result? There's a quiescent period, right? This is actually pretty cool. Know the time of the quiescent period. What is the quiescent period? You say that 10 times fast enough, you're going to quiescent period, right? It's the actual, nothing's happened. There's no contraction. There's no, the heart is filled, and it's a really cool, it's an interesting concept. So check it out. Make sure you know what it means. Nodal rhythm. Well, what is a nodal rhythm versus a non-nodal rhythm? Think about that. Again, it goes back to the SA node and AV node. Be familiar with the blood that flows through the heart and all the associated structures. Like if you were going to begin at the superior and inferior vena cava, blood enters the right atrium, goes through the valve, to the right ventricle, out to the pulmonary aortic, right? Comes back to the pulmonary veins, to the left atrium, mitral valve, right ventricle, aortic semilunar valve, to the aorta, right? So if you were to treat, obviously that's not how it works because it's a simultaneously beating chambers, right? The atriums contract together and the ventricles contract together. But if you had to trace blood through the heart, just pick one spot and go through it and be able to name all the sections, all the spots. When you get in lab, that's what you're going to do. You're going to find that big mile of the heart. You're going to start at the superior vena cava, inferior vena cava, and work your way to the pulmonary side of the heart. Remember, the heart is it's a pump. It's a two-part pump. One part is pulmonary, which goes to the lungs, and the other part is systemic, going to the body. So when you start on the pulmonary side, which is the right side, you are bringing deoxygenated blood to the heart, pumping into the lungs, bringing it back to the systemic side with oxygen, pumping it out to the body, right? Systemic is body, pulmonary is the lungs, right? And the good thing is, right, if we compare the left ventricle and the right ventricle is not as thick as the left ventricle because the right ventricle has to pump blood to the lungs, and the lungs are within proximity of the heart. It's like right next to it, right? As opposed to pumping blood to your toe from the left side of the heart, you need a thicker ventricle to get that ejection out. My little video's in the way again, but ECGs, electrocardiograms, be familiar with your P wave, QRS, T waves, normal versus abnormal. What does it mean not to have a P wave or a T wave? QRS. Well, and then I want you to take the concept of the electrocardiogram, P wave, atrial filling, right? What do you think? And then QRS, and then T, repolarization. Take that electrocardiogram and compare it to the chart that's in the book on the repolarization and depolarization of each of the chambers during as it correlates with the P wave, QRS, and T wave. So take a normal section of the ECG, and it's compared for you in the textbook, and see the relationship between the depolarization and repolarization of the actual heart muscle compared to the ECG. It's very, very interesting. Cardiomyocytes, basically your cardiac cells. Why is a cardiac cell so special? What's the structure? What does it contain that makes it so special? How does a heart not blow apart every time it contracts? That's just one of the special structures and functions of the cardiomyocytes. Some definitions: preload, afterload, stroke volume, cardiac output, ejection fraction. There is a little. I'm pretty sure there is a little how to calculate cardiac output with heart rate versus stroke volume. Okay, but these are some definitions you should be familiar with. Again, I just said this before, but your ECG in relation to systole and diastole, meaning depolarization and repolarization. It's a really good correlation between that P wave, QRS, and T wave and the actual motion of the heart. Some other definitions and locations: cardiac center versus cardioinhibitory center versus baroreceptors or baroreceptors. What's their function? Where are they located? What's their role? Why can you remove the heart from the body and it still beats on the table? In a rhythm. Is it a nodal rhythm? Why can you take the heart and cut it into 4 separate chambers and it still beats or pulsates? Right down to the individual cell and its ability to depolarize on its own. The heart is very special. Definition: diastasis versus isovolumetric contraction. The isovolumetric contractions in that chart, which kind of compares again the ECG with repolarization and depolarization. Check it out. Heart linings, pretty straightforward. What are the 3 heart linings and the histology? What makes them up? Where are they located? Who's on the outside? Who's in the middle? Who's on the inside? Right? A little hint. Right? The one that starts with the M is in the middle. M, myocardium. Myo means muscle. That's the bulk of your heart, right? So in the middle is the myocardium. Endo is in, and then ecto is out, right? Which you already knew. Some definitions. I spelt it wrong. I apologize. Positive ionotrophic effect. This should be I-O-N, trophic effect. Positive versus negative chronotrophic effect. Again, I apologize. So over here is positive ionotrophic effect, not inotrophic effect, but negative chronotrophic effect. What does that mean, and where is it going? Or definitions. Again, the heart is myogenic. What does that mean? What does it mean to be myogenic? I think I just explained it. Fibroskeleton. It should be in the textbook because the fibroskeleton is one of the most important parts of the heart itself. You take the heart and strip away all the muscles, all the little other tiny structures like your heartstrings and your papillary muscles. You're left with this fibrous beings, fibrous skeleton. The fibrous skeleton is very important. If the heart works with electrical impulses, it's this fibrous skeleton that prevents the rogue impulse from going where it doesn't need to go. It kind of focuses the energy, if you would, from the SA node to the AV node to the bundle of His, so forth and so on. Purkinje fibers, a bundle of His, or bundle of His to the Purkinje fibers. But the fibrous skeleton does more than that. It is your ability to retract and retract. It also helps prevent the explosion of your heart every time you contract it. Pericarditis, friction rub. What does that mean? What are some symptoms of pericarditis? Where is it located? Valves and all their sounds, and valve conditions, like mitral valve prolapse, like mitral valve stenosis. What is a prolapse versus stenosis? Lub versus dub, right? Your S1, your S2. Is there an S3, S4 sound? Absolutely. Right? So there's clicks, there's a drawn out S2 sound, or there's a shorter S1 sound, all different things based on pathology of the heart, which you don't have to know about. But the textbook does cover valves, some conditions that affect those valves, and of course your lub and your dub. What are those sounds there? Looking at the physiology a little bit more, receptors, acetylcholine, nicotinic, muscarinic receptors, what are they? Who attaches to them? What's the effect of epinephrine? What's the effect of acetylcholine on these receptors? How do you speed the heart up? How do you slow it down? Again, ionotrophic effect versus chronotrophic effect. What's the vagus nerve? What's the vagus nerve influence on the heart? If you didn't have the vagus nerve attached to the heart, your heart would beat at a regular beat of 120 beats per minute, straight out flat. That's exactly what it's set at. Your heart is already have an intrinsic ability to beat at 120, 110, 120 beats per minute without the vagus nerve. Your vagus nerve is your brake. It is what slows your heart down. It brings it back to a good rhythm. It is your brake. Without the vagus nerve, and you take the heart out of the body and slap it on the table, it's going to beat 120 beats per minute, give or take. You plug in the vagus nerve and it slows it down. Absolute refractory period. The absolute refractory period, what does that mean? Again, it relates back to your ability to depolarize and repolarize the heart and its effect with the ECG. And lastly, a little hint, okay, the left ventricle is pressurized at 120 millimeters of mercury, whereas the right ventricle is pressurized at a maximum of 25 millimeters of mercury, meaning it takes 120 millimeters of pressure to get that left ventricle to contract. And why is that number higher? Because the muscle's thicker. The muscle's thicker on the left side because it has to reject the blood with oxygen to the systemic side, to the systemic body, right? It's pretty much the systemic system on that left side. The right side doesn't take much pressure because it's a thinner wall ventricle. It doesn't take much pressure to get from the right ventricle to the lungs because of the proximity of the lungs. So I guess these numbers here I'm showing you, this is an actual test question. The test question is, what's the pressure on? Left ventricle is pressurized at 120, meaning it's not blood pressure. It's that what is it pressurized at? And it's how much pressure does it take to eject blood from the left ventricle? That's 120 millimeters of mercury. It takes more because you're sending the blood to the body. It doesn't take much to pressurize the right ventricle or to eject the blood to the lungs because the lungs are right next to your heart. So when you see that question, the answer is 120 over 25, or it's actually 120 left ventricle, 25 right ventricle. It has nothing to do with blood pressure whatsoever. What is normal blood pressure, right? 140, what's the normal high? 140 over 80, times are changing. Times are changing. That number might be different now. It might be 140 over 90. When I was in school, we learned 140 over 80. 140 over 90 was borderline high. All right, so that wraps up chapter 19. There's a lot of information here in chapter 19, a lot of great information if you just put it together and understand the function of the heart, right? The form and function. Why is it formed the way it is versus the function of what it does? Any questions, concerns, or comments, please feel free to contact me. And I'm here to help and enjoy chapter 19.